Chapter 9: Immunity mediated by B cells and antibodies. B cell activation requires cross-linking of surface immunoglobulin. B cell activation begins when antigens on a bacterial cell bind to and cross-link multiple B cell receptors, or BCRs, on the surface of a B cell. This clustering of antigen-bound receptors brings together intracellular signaling molecules. Specifically, receptor-associated kinases, like BLK, FYN, and LIN, are activated and begin phosphorylating the items, or immunoreceptor tyrosine-based activation motifs, located on the cytoplasmic tails of the BCR complex. Next, the kinase Syk is recruited. It binds to the doubly phosphorylated items, and upon binding, Syk itself becomes activated. This triggers a cascade of intracellular signals, ultimately resulting in changes in gene expression in the nucleus. These gene expression changes prepare the B cell to proliferate and produce antibodies. This precise and regulated signaling ensures that B cells are only activated when they encounter specific antigens, keeping the immune response both targeted and efficient. B cell activation requires signals from the B cell co-receptor. In addition to antigen binding, B cell activation also requires input from the B cell co-receptor. This co-receptor complex is made up of three key components: CD19, CD81, and CR2. Here's how it works. When a pathogen is opsonized with C3b, a complement protein, CR1 on the B cell surface binds to C3b. This binding recruits factor I, which cleaves C3b into I3b and then into C3d. Once this happens, the CR2 component of the B cell co-receptor binds to C3d on the pathogen surface. This dual engagement, antigen binding to the B cell receptor and complement binding to the co-receptor, amplifies the signal and ensures full B cell activation. Together, these pathways allow the B cell to respond rapidly and specifically to invading pathogens. B cell activation is most effective when the B cell receptor, or BCR, works together with the B cell co-receptor complex. In the scenario on the left, we see a B cell interacting with a bacterial pathogen. The antigen on the pathogen binds to the membrane-bound IgM on the BCR. At the same time, the complement protein C3d, which is attached to the pathogen, binds to the CR2 component of the co-receptor complex. This complex also includes CD19 and CD81. In the right panel, the same cooperative signaling happens, but with a soluble antigen. The antigen is opsonized with C3d, which again binds CR2, while the antigen itself engages the BCR. In both cases, this dual engagement results in synergistic activation signals. These signals are stronger and more efficient than activation through the BCR alone. As a result, downstream signaling cascades are amplified, leading to significant changes in gene expression in the B cell nucleus, preparing the B cell for proliferation, differentiation, and antibody production. Effective B cell-mediated immunity depends on help from CD4 T cells. While most effective B cell responses rely on help from CD4 T cells, some antigens can activate B cells independently. This process is called B cell activation by a thymus-independent antigen. In this example, a bacterial surface is densely coated with repetitive antigenic epitopes. These epitopes simultaneously engage and cross-link multiple B cell receptors. At the same time, the B cell co-receptor complex, including CR2, CD19, and CD81, binds to complement components like C3d that are also attached to the pathogen. This powerful clustering of BCRs and co-receptors delivers a strong enough signal to activate the B cell, without the need for T cell involvement. Although this pathway leads to activation, the response is generally weaker and shorter-lived than a T-cell dependent response, and it typically doesn't produce strong memory or class switching. Follicular dendritic cells in the B-cell area store and display intact antigens to B cells. What you're seeing here is a high-resolution electron micrograph of a follicular dendritic cell, or FDC, located in the B-cell zone of a lymphoid follicle. The central mass is the FDC cell body, while the surrounding long, thin processes extend outward like delicate branches. These processes are studded with immune complexes, including intact antigens, which the FDC stores and displays to B cells over long periods. Unlike classical dendritic cells, follicular dendritic cells do not process antigens. Instead, they retain whole antigens on their surface, tethered via complement or antibody molecules. This prolonged antigen presentation is essential for B-cell selection and survival during the germinal center reaction, helping shape long-lasting and high-affinity antibody responses. Let's take a closer look at how follicular dendritic cells, or FDCs, capture and display intact antigens in the B-cell zone of lymph nodes. When a pathogen enters through the afferent lymphatic vessel, it carries antigens tagged with complement proteins like C3d. These antigens travel to the subcapsular sinus, where subcapsular sinus macrophages and CR2-expressing B cells capture them. Some antigens are transferred to B cells and guided into the primary follicle, where FDCs reside. These FDCs express complement receptors, especially CR2, which allow them to bind and retain intact antigens for extended periods. Inside the follicle, the FDC's branching processes present these antigens to naive B cells. This interaction is essential for B cell selection, affinity maturation, and the development of memory cells in the germinal center. In this way, FDCs create a localized and sustained antigen reservoir, ensuring B cells receive the necessary signals to fine-tune the antibody response. Antigen-activated B cells move close to the T cell area to find a helper TFH cell. Once B cells are activated by antigen and co-receptor signaling, their next step is to seek help from T follicular helper. In the first panel, naive B cells migrate through the lymphoid follicle, scanning follicular dendritic cells for specific antigens. At the same time, naive T cells in the T cell zone are engaging conventional dendritic cells, which present processed peptide antigens. When both B and T cells recognize the same antigen, they are activated in parallel. This sets the stage for collaboration. In the middle panel, activated B cells begin to migrate toward the boundary region between the B cell and T cell areas, while activated T cells also proliferate and move in the same direction. Finally, in the third panel, antigen-activated B cells present antigen-derived peptides via MHC class II to T cells. This leads to the formation of cognate BT pairs, enabling the T cells to deliver essential cytokine and co-stimulatory signals to fully support B cell differentiation. This critical interaction drives the formation of germinal centers and long-lasting, high-affinity antibody responses. Once an antigen-activated B cell encounters a matching T follicular helper cell, a highly coordinated process begins to fully activate the B cell. In the first step, the T cell forms a stable conjugate with the B cell. This interaction is stabilized by surface molecules like ICAM1 and LFA1, and it triggers the T cell to begin synthesizing cytokines and CD40 ligand. Next, the T cell reorients its cytoskeleton and Golgi apparatus so that its secretory machinery points directly at the B cell. This ensures that the activation signals are precisely targeted. In parallel, The cytoskeletal protein talon concentrates at the zone of cell contact, clearly visible in the fluorescent micrograph on the right. Finally, cytokines produced by the T cell are secreted into the narrow synaptic space between the two cells, delivering the final signals the B cell needs for full activation, proliferation, and differentiation. This intimate interaction between B and T cells is essential for generating long-lasting immunity and robust antibody production. The primary focus of clonal expansion in the medullary cords produces plasma cells secreting IgM. After receiving help from a TFH cell, activated B cells begin to proliferate rapidly, a process known as clonal expansion. In the first panel, the primary focus of this expansion takes place in the medullary cords. Here, B cells differentiate into early plasma cells that secrete IgM antibodies, providing an initial wave of protection. Meanwhile, other activated B cells migrate back into the primary follicle, forming the secondary focus of expansion. These cells continue dividing and interacting with follicular dendritic cells. In the third panel, this expansion in the follicle gives rise to a germinal center, where B cells undergo somatic hypermutation and affinity maturation. This process helps generate long-lived plasma cells and memory B cells that produce high-affinity antibodies. Together, the primary and secondary foci ensure that the immune response is both rapid and refined, starting with broad IgM production and progressing to specialized, long-lasting protection. This electron micrograph shows a plasma cell, the final product of B cell differentiation following activation and clonal expansion. At the center of the image is the large, dense nucleus, labeled N, which is characteristic of plasma cells actively engaged in transcription. Surrounding it, we see an abundance of rough endoplasmic reticulum, or RER, clearly labeled at the bottom of the image. This extensive RER is a hallmark of plasma cells, as it reflects the high levels of protein synthesis required to produce and secrete large amounts of antibodies, especially IgM in early immune responses. The structure of this cell confirms its role as a dedicated antibody factory, providing the first wave of humoral defense during an active immune response. Activated B cells undergo somatic hypermutation and isotype switching in the specialized microenvironment of the primary follicle. This figure shows the highly organized microenvironment of the germinal center, where activated B cells undergo somatic hypermutation and isotype switching. In the schematic on the top left, we see two main functional areas within the germinal center: the dark zone, packed with rapidly dividing centroblasts, and the light zone, where centrocytes interact with follicular dendritic cells and T cells for selection. The surrounding mantle zone contains resting B cells, while the T cell zone is adjacent to support continued T cell health. To the right, we see an immunofluorescent stain of a lymph node section. The dark zone appears green, the light zone red, and the mantle zone blue, confirming this compartmentalization. Below, the light micrograph on the left shows dense populations of cells in the germinal center, and the image on the right specifically stains for follicular dendritic cells, highlighting their extensive network. This specialized structure supports the refinement of the antibody response, selecting for high-affinity B cell clones while also guiding isotype class switching. Antigen-mediated selection of centrocytes drives affinity maturation of the B cell response in the germinal center. Inside the germinal center, B cells undergo somatic hypermutation, generating variations in the affinity of their surface immunoglobulins. These mutated B cells, now called centrocytes, must compete for survival signals. In the left panel, a centrocyte with low affinity immunoglobulin fails to bind antigen effectively. As a result, it cannot present antigen to a TFH cell. Without this interaction, the centrocyte receives no survival signals and undergoes apoptosis. On the right, a centrocyte with high affinity surface immunoglobulin successfully binds antigen. This antigen is processed and presented on MHC class II to a TFH cell. If the interaction is strong and CD40-CD40L signaling occurs, the centrocyte receives help in the form of cytokines and survival signals, such as BCL-XL. These selected centrocytes then either differentiate into plasma cells, which produce high-affinity antibodies, or re-enter the cycle for further rounds of mutation and selection. This process, known as affinity maturation, ensures that only the most effective B cells are preserved, driving the quality of the antibody response forward. The cytokines made by helper T cells determine how B cells switch their immunoglobulin isotype. B cells don't just make antibodies, they can switch the isotype of the antibodies they produce based on signals received from helper T cells, particularly in the form of cytokines. This figure summarizes how different cytokines influence antibody isotype switching. IL-4 inhibits IgM, IgE3, and IgE2, but induces IgE1 and IgE, making it key for allergic responses and parasitic defense. IL-5 plays a more limited role, but it augments the production of IgA, supporting mucosal immunity. IFN-gamma inhibits IgM and IgE, but induces IgE3 and IgE2, which are important for viral and intracellular bacterial defense. TGF-beta suppresses IgM, IgE3, and IgE2, but induces IgE2b and IgA, making it critical for immune tolerance and mucosal protection. These cytokine signals come from TFH cells during cognate-BT interactions, directing B cells to produce the most appropriate antibody for the type of pathogen encountered. Together, this cytokine-driven switching tailors the immune response for specific pathogens and tissues. This figure highlights the functional consequences of failed isotype switching by comparing two lymph node samples. The top panel shows a lymph node from a patient with hyper-IgM syndrome. This genetic disorder is characterized by defective CD40-CD40L interactions, preventing B cells from receiving the necessary signals to undergo class switching and germinal center formation. As a result, the lymph node appears disorganized, and germinal centers are completely absent. In contrast, the bottom panel shows a normal lymph node, where multiple clearly defined germinal centers are visible. These structures represent areas of active B cell proliferation, somatic hypermutation, and selection, key to generating high-affinity, isotype-switched antibodies. This comparison illustrates just how essential T-cell help and cytokine signaling are for mounting an effective adaptive immune response. Cytokines made by helper T cells determine the differentiation of antigen-activated B cells into plasma cells or memory cells. Once B cells have been selected for high-affinity receptors in the germinal center, they face a critical decision whether to become plasma cells or memory B cells. This decision is largely influenced by cytokines secreted by TFH cells. In the left panel, an IL-10 secreting TFH cell interacts with a centrocyte through CD40-CD40L binding. In the presence of IL-10, the centrocyte is signaled to differentiate into a plasma cell. These cells are the body's antibody factories, producing large quantities of immunoglobulin to help eliminate the current infection. In the right panel, another centrocyte, bearing an identical B cell receptor, receives help from a TFH cell that secretes IL-4. This cytokine promotes the formation of memory B cells, which do not secrete antibodies immediately but persist long-term. These memory cells serve as a lasting investment, enabling a faster and stronger immune response upon future exposure to the same pathogen. Thus, TFH-derived cytokines help direct the strategic allocation of B-cell resources, balancing immediate defense with long-term protection. This chart highlights the key functional differences between naive B cells and plasma cells across both intrinsic and inducible properties. Naive B cells express surface immunoglobulin and MHC class II, allowing them to recognize antigen and present it to T cells. However, they do not secrete antibodies at high rates. In contrast, plasma cells have lost both surface IgE and MHC class II expression, but they are highly specialized for antibody secretion, producing large quantities of immunoglobulin to neutralize pathogens. When it comes to inducible properties, naive B cells are still flexible. They can undergo growth, somatic hypermutation, and isotype switching in response to activation and T cell help. Plasma cells, however, are terminally differentiated. They are locked into their role, no longer growing, mutating, or switching antibody classes. In summary, this table underscores the developmental trade-off. Naive B cells are versatile responders, while plasma cells are dedicated effectors. IgM, IgE, and monomeric IgE protect the internal tissues of the body. This figure explains how IgE antibodies are transported from the bloodstream into internal tissues via FcRn, the neonatal Fc receptor. In the first panel, fluid phase endocytosis allows endothelial cells to take up IgE from the blood. Inside the endocytic vesicle, the acidic pH triggers IgE to bind to FcRn. In the second panel, this binding protects the IgE molecule from being degraded in the acidic vesicle environment. Then, as the vesicle moves toward the basolateral surface of the endothelial cell, the pH shifts. In this more basic environment, shown in the third panel, IgE is released from FcRn into the extracellular space. This pathway ensures that IgE antibodies are preserved and delivered to sites where they're needed for immune protection, without being destroyed during transport. It's one of the key ways that the immune system extends systemic antibody protection into deeper tissues. Dimeric IgA protects the mucosal surfaces of the body. This figure illustrates how dimeric IgA is transported across epithelial cells to protect mucosal surfaces. In the first panel, IgA is secreted by plasma cells located in the lamina propria. It binds to the polymeric IgE receptor, or PIGR, on the basolateral surface of epithelial cells. In the second panel, this complex is taken into the epithelial cell via receptor-mediated endocytosis. In the third panel, the IgE PIGR complex travels through the cytoplasm and is delivered to the apical surface of the epithelial cell, where it prepares for release. Finally, in the fourth panel, the receptor is cleaved, releasing IgA into the mucus layer. This process leaves behind a piece of the PIGR known as the secretory component, which remains bound to IgA. This component stabilizes the antibody and protects it from enzymatic degradation. The end result is a dimeric IgA molecule embedded in mucus, forming the first line of immune defense at mucosal barriers such as the gut, lungs, and reproductive tract. IgE provides a mechanism for the rapid ejection of parasites and other pathogens from the body. This figure shows how IgE provides a powerful defense mechanism for the rapid ejection of parasites and other pathogens. In the left panel, we see an inactive mast cell filled with preformed granules that contain histamine and inflammatory mediators. These mast cells express high-affinity Fc-epsilon-RI receptors, which are loaded with IgE antibodies even in the absence of antigen. When multivalent antigen is introduced, as shown in the right panel, it binds and cross-links the surface-bound IgE. This triggers mast cell activation. As shown in the upper right electron micrograph and lower diagram, cross-linking leads to immediate degranulation, a rapid release of histamine and other pro-inflammatory substances stored in granules. This powerful response results in increased vascular permeability, smooth muscle contraction, and mucosal secretion, physiological changes that help expel parasites or allergens from the body. Though this pathway is essential for parasite defense, it is also responsible for allergic reactions, such as hay fever, asthma, and anaphylaxis. This figure highlights the critical role of eosinophils in IgE-mediated defense against large multicellular parasites, such as schistosomes. In the upper panel, we see that eosinophils express Fc epsilon Ri, the high-affinity receptor for IgE. When IgE antibodies bind to antigens on the surface of a parasite, like a schistosome, they anchor eosinophils directly to the pathogen. This binding triggers the release of cytotoxic granules from the eosinophils, containing enzymes and proteins designed to damage and destroy the parasite's outer membrane. The bottom panel shows a microscopic image of a schistosome larva cell surrounded by eosinophils, labeled E. The eosinophils cluster tightly around the parasite, indicating an active immune assault. This process is a hallmark of the type 2 immune response, specialized for targeting pathogens too large to be phagocytosed. Together, IgE and eosinophils provide a potent line of defense against parasitic infections that threaten mucosal and systemic tissues. Mothers provide protective antibodies to their young, both before and after birth. This final figure shows how maternal antibodies protect infants both before and after birth, offering essential passive immunity during early life. In the first panel, we see that various antibody isotypes, including monomeric IgA, IgE, dimeric IgA, and IgE, are produced at different mucosal and systemic sites in the mother's body. In the second panel, IgE is actively transported across the placenta to the fetus via FcRn receptors, giving the baby systemic protection against pathogens during gestation. After birth, as seen in the third panel, the mother continues to provide immune protection through breastfeeding. Here, dimeric IgA is secreted into breast milk and transferred to the infant's gastrointestinal tract. This antibody coats mucosal surfaces, neutralizes pathogens, and prevents microbial invasion. Together, transplacental IgE and secretory IgA from breast milk form a powerful immune shield that protects the newborn during the vulnerable first months of life, before their own immune system fully develops. This graph illustrates how antibody protection evolves from conception through early childhood, highlighting the critical role of maternal IgE and the gradual rise of the child's own antibody production. During pregnancy, maternal IgE is transferred across the placenta, peaking around the time of birth. This gives the newborn immediate systemic protection, but this passive immunity is temporary. After birth, maternal IgE levels rapidly decline, creating a transient window of vulnerability, marked here in blue, where IgE levels dip before the infant's immune system ramps up. Starting around birth, the infant begins producing its own IgM, followed by IgE and IgA. Over the next few years, these antibody levels gradually increase. By ages 5 to 6, the child's serum immunoglobulin profile approaches that of a healthy adult. This progression underscores the importance of maternal antibodies during infancy, while also highlighting the need for vaccination and nutritional support as the child builds their own adaptive immune response. High-affinity neutralizing antibodies prevent viruses and bacteria from infecting cells. This figure shows how high-affinity neutralizing antibodies, particularly IgA at mucosal surfaces, can prevent viral infections, using influenza as an example. On the left side, an adult with pre-existing anti-influenza IgA antibodies encounters the virus at the respiratory epithelium. The IgA binds to the virus, blocking its ability to attach to and enter host cells. As a result, the virus cannot replicate, and the individual remains healthy. On the right side, her twin sister lacks these antibodies. The influenza virus successfully binds to epithelial receptors, enters the cell, and begins replicating. Without neutralizing IgA, the virus spreads, causing severe illness for 2 weeks. This comparison underscores the power of neutralizing antibodies in preventing infection before it begins, especially at mucosal entry points like the respiratory tract. Such antibodies are often the goal of effective vaccination, especially for respiratory and gastrointestinal pathogens. This figure shows how high-affinity IgA antibodies help control bacterial colonization in the upper respiratory tract, specifically against Streptococcus pyogenes in the pharynx. On the left, a child with pre-existing IgA antibodies against S. pyogenes encounters the bacteria. The IgA binds to the pathogen's F protein, blocking its ability to adhere to fibronectin on epithelial surfaces. As a result, the bacteria are swept away by the cilia and directed into the gut for clearance. The bacterial load remains controlled and the child stays healthy. On the right, the child's brother lacks these protective antibodies. Without IgA to block adhesion, the bacteria firmly attach to epithelial surfaces, resist clearance, and begin to replicate. The growing bacterial population triggers inflammation, leading to sore throat symptoms. This example illustrates how IgA acts as a neutralizing barrier, not just against viruses, but also against bacteria by preventing them from ever attaching and infecting host cells. High-affinity IgE and IgA antibodies are used to neutralize microbial toxins and animal venoms. This table summarizes how high-affinity IgE and IgA antibodies protect us by neutralizing microbial toxins and venoms, which can cause severe disease even at low concentrations. Each row shows a specific disease, the organism responsible, the toxin involved, and the effect it causes in the body. For example, tetanus toxin from Clostridium tetani blocks inhibitory neurons, causing sustained muscle contraction. Diphtheria toxin inhibits protein synthesis, leading to myocarditis and cell damage. Botulinum toxin causes paralysis by blocking acetylcholine release. Toxins like cholera, pertussis, and anthrax disrupt immune or epithelial barriers, while others, like streptolysin and scarlet fever or enterotoxins in food poisoning, cause inflammation or direct tissue injury. Neutralizing antibodies bind these toxins before they interact with host cells, blocking their toxic effects. Vaccines for diseases like tetanus, diphtheria, and pertussis work by generating these high-affinity antibodies. Ultimately, antibody-mediated neutralization is one of the most effective defenses in the immune arsenal, especially when toxins are the main drivers of disease. This figure demonstrates how neutralizing antibodies protect cells by blocking the entry and action of microbial toxins. In the first 3 panels, we see the typical path of toxin-mediated cell damage. First, the toxin binds to a cell surface receptor. Then, it's taken into the cell through endocytosis. Inside, the toxin dissociates and releases its active chain, which poisons the cell by disrupting critical processes. In the 4th panel, we see how antibodies prevent this from happening. Neutralizing antibodies bind directly to the toxin, blocking its ability to interact with the receptor in the first place. By preventing initial binding, the entire toxic pathway is stopped before it starts, making IgE and IgA antibodies powerful defenders against microbial toxins and venoms. Binding of IgM to antigen on a pathogen surface activates complement by the classical pathway. This chart ranks antibody isotypes based on their ability to fix complement through the classical pathway, an essential mechanism for tagging and destroying pathogens. At the top, IgM and IgE are the most potent activators, both marked with +++ due to their structural efficiency in binding C1q, the first complement protein. IgE1 also fixes complement effectively, while IgE2 and IgA subclasses have more limited capacity. In contrast, IgE, IgE4, and IgE do not activate complement at all. Instead, these isotypes play specialized roles, such as IgE in allergy and parasitic defense, or IgE4 in immune tolerance. Understanding the complement-fixing abilities of each antibody class helps explain their biological roles and informs therapeutic antibody design. This figure details how IgM activates the classical complement pathway once it binds to a pathogen surface. IgM is first secreted in a planar form, which cannot bind complement. But upon binding to multiple antigens on a pathogen surface, it adopts a staple conformation, exposing sites for C1 complex binding. The C1 complex initiates the cascade by activating C2 and C4, forming C4b2, the classical C3 convertase. This convertase cleaves C3 into C3a and C3b. C3a acts as an anaphylatoxin, promoting inflammation. C3b binds to the pathogen surface, tagging it for phagocytosis or driving the formation of the C5 convertase, leading to pathogen lysis by the membrane attack complex. This cascade amplifies the immune response, enhancing clearance and destruction of the pathogen through complement-mediated immunity. In short, IgM is the most efficient initiator of classical complement activation, turning early humoral recognition into a powerful effector mechanism. This final figure highlights the amplification of complement activation on the surface of a pathogen, initiated by antibody binding. In the first panel, C1 binds to the antigen-antibody complex, usually IgM or IgE3, triggering the classical pathway. Next, C4b is deposited by the activated C1 complex. This helps form the classical C3 convertase, C4b2. In the third panel, C4b2 cleaves C3, depositing multiple C3b molecules on the surface. This tagging is critical for opsonization and the next phase of the response. Finally, the figure shows that C3b can also participate in the alternative pathway, forming C3bbb, an alternative C3 convertase that amplifies the deposition of even more C3b. This positive feedback loop rapidly escalates the response, coating the pathogen with complement proteins that facilitate phagocytosis and destruction. Two forms of C4 tend to be fixed at different sites on pathogen surfaces. This figure illustrates the genetic variation of C4 genes, showing how individuals inherit different combinations of C4a and C4b. Each horizontal line represents the C4 gene configuration in a different person. The yellow blocks represent C4a genes, and the blue blocks represent C4b genes. C4a and C4b encode functionally distinct isoforms of the C4 protein. C4a tends to bind to amino groups, favoring protein-rich surfaces. C4b tends to bind to hydroxyl groups, favoring carbohydrate-rich surfaces. This diversity allows the complement system to adapt to a wide variety of microbial surfaces, increasing its ability to label pathogens for clearance. The distribution of these gene variants varies between individuals, contributing to immune system variability and potentially influencing susceptibility to infection or autoimmune disease. Complement activation by IgE requires the participation of two or more IgE molecules. This figure shows how IgE activates complement, but only when two or more IgE molecules are brought together by antigen binding. On the left, IgE antibodies bind to antigens on a bacterial surface. When enough antibodies are closely spaced, they provide a platform for C1q to bind. This clustering triggers the classical complement pathway, leading to pathogen destruction. On the right, a similar mechanism occurs with soluble multivalent antigens. Here, IgE molecules form an immune complex, which also allows C1q binding and complement activation. The key takeaway is that a single IgE molecule is not enough. Complement activation requires antibody clustering to recruit and activate C1q. This requirement ensures that complement is only triggered when there's a real threat, minimizing the risk of damaging healthy tissues. Erythrocytes facilitate the removal of immune complexes from the circulation. This figure explains how erythrocytes play a key role in immune homeostasis by removing circulating immune complexes from the bloodstream. At the top, we see a small antigen-antibody complex forming in the blood. Once formed, it activates complement, leading to the deposition of C3b on the complex. In the third panel, CR1 receptors on the surface of an erythrocyte bind the C3b tagged immune complexes. The erythrocyte then travels through the bloodstream and carries these bound complexes to the liver or spleen. Finally, at the bottom, a resident macrophage detaches the immune complexes from the erythrocyte and phagocytoses them for safe disposal. This mechanism prevents immune complexes from depositing in tissues, helping to avoid inflammation and damage associated with conditions like lupus or glomerulonephritis. Fc receptors enable effector cells to bind and be activated by IgE-bound pathogens. This figure illustrates how Fc gamma receptor I, or Fc gamma RI, allows effector myeloid cells to recognize and respond to IgE-bound pathogens. In the first panel, Fc gamma RI is shown on a myeloid cell membrane. It's made of an alpha chain, which binds IgE, and a gamma chain, which transduces activation signals. The second panel shows that Fc gamma RI binds to the lower hinge and CH2 domain of IgE3, the isotype with the longest hinge region and strongest binding affinity. Finally, when the Fc gamma RI IgE3 complex encounters antigen, it triggers activation of the effector cell. This activation can lead to phagocytosis, cytokine release, or respiratory burst, depending on the myeloid cell type, making Fc gamma RI crucial in antibody-dependent cell-mediated immunity. This final figure illustrates how Fc receptors on macrophages mediate the phagocytosis of antibody-coated bacteria. In the first panel, antibodies bind to a bacteria. The Fc region of these antibodies is recognized by Fc receptors on the surface of a macrophage. Once bound, the receptors transmit activating signals to the macrophage. The cell then extends its membrane around the bacteria, forming a phagocytic cup. The membranes fuse, creating an internal vesicle called a phagosome. In the last step, the phagosome fuses with a lysosome, forming a phagolysosome, where digestive enzymes and reactive species kill and degrade the pathogen. This process, called antibody-dependent phagocytosis, links the adaptive immune response, via antibodies, to the innate effector functions of professional phagocytes. A variety of low-affinity Fc receptors are IgE-specific. This figure summarizes the major Fc gamma receptors that recognize IgE and regulate immune responses. On the top row, we see the receptor names: Fc gamma Ri, Fc gamma Ria, Fc gamma RIIB1 and 2, Fc gamma Ria, and Fc gamma Reeb, also known by their CD markers like CD64, CD32, and CD16. Below that, their structures differ by molecular weight and cytoplasmic signaling domains. Some receptors have items which promote activation, while others, like Fc gamma Reeb, contain items which inhibit signaling. Next, we see subclass specificity. Fc gamma Ri binds Ig3 best, while Fc gamma Ria varies depending on genetic polymorphisms like R131 and H131. Their relative affinity for Ig1 also varies. Fc gamma Ri binds with the highest strength, while others show much lower affinity, often requiring immune complex formation to signal. Finally, we distinguish their functional outcomes. Some promote phagocytosis or ADCC, while others downregulate the immune response to prevent overactivation. Together, these receptors finely tune the immune response, balancing activation and inhibition based on antibody engagement. An Fc receptor acts as an antigen receptor for NK cells. This figure illustrates how Fc receptors on natural killer cells enable them to kill antibody-coated target cells, a process known as antibody-dependent cellular cytotoxicity, or ADCC. In the first panel, an anti-CD20 antibody binds to the CD20 protein on the surface of a B-cell lymphoma cell. Next, Fc gamma receptor 3, or CD16, on the surface of an NK cell, recognizes and binds the Fc region of the antibody. When enough Fc receptors are cross-linked, the NK cell becomes activated. The NK cell then releases cytotoxic granules, triggering apoptosis in the B-cell lymphoma. This mechanism is critical in the immune response and is also leveraged in antibody-based cancer therapies like rituximab. The Fc receptor for monomeric IgA belongs to a different family than the Fc receptors for Ig and IgE. This chart summarizes the structure and cellular distribution of Fc receptors for Ig, IgE, and IgA. Starting on the left, the Fc gamma receptors for Ig differ in structure, function, and expression. Some are activated, like Fc gamma Ri and Ria, while others, such as RIIB2 and RIB1, serve inhibitory roles. These receptors are expressed across a variety of immune cells, including macrophages, neutrophils, dendritic cells, and even platelets and NK cells. Next, the Fc epsilon receptor I, which binds IgE, is a powerful activating receptor found on mast cells, basophils, and eosinophils, key players in allergic responses. Finally, the Fc alpha receptor I, or Fc alpha Ri, is the only IgA-specific Fc receptor shown here. It is encoded on chromosome 19 and is found on macrophages, neutrophils, eosinophils, and dendritic cells. These differences explain how each Fc receptor tailors immune responses based on the type of immunoglobulin involved.